This is a review of the lesson reviews for Chapter 4. I'm going to go kind of quickly. Obviously, you can pause this if you need to, but the intention here is to review the answers that you already have written down in your notebooks. It is not meant for you to copy them because you are supposed to do them on your own. This is just so you can check your answers. All right, 4.1 was the first one you had to do. The main thing that you need to know going forward is genetics is the study of inheritance. Inheritance being what you get passed down genetically from your parents. Mendel chose to work with pea plants because they had very obvious trait differences. The pea was either wrinkled or it was smooth. There was no in between. So he could very easily tell which trait was showing up in that pea plant. He was one of the first ones to show that there was no blending. Previous to Mendel, they thought if you had a really tall person and a really short person, that those traits would blend and you would get an average sized person. And then one of the interesting things is he saw that if he took two plants that always produced the same kind of peas, that they were always wrinkled, and then he cross-pollinated those, then they would produce all wrinkled seeds. But then if he allowed two of those seeds to grow and then cross-pollinated those, then he ended up with some that were wrinkled and some that were smooth. So he had to explain that, and that's where he came up with the idea of dominant and recessive traits. All right, 4.4 was on DNA. There's a little bit more meat here that you need to be aware of. DNA is a two-stranded molecule. It has two strands that are twisted around each other like a double helix or a spiral staircase, or imagine a ladder and then you twist it. Okay, that's kind of what they look like, with the nitrogen bases making up the rungs of that twisted ladder. Okay. Speaking of those nitrogen bases, they're part of the nucleotides that make up DNA and RNA. Each nucleotide has a phosphate group and a 5-carbon sugar. In DNA, that 5-carbon sugar is deoxyribose. In RNA, it's ribose. That's why it's DNA and RNA. And then nitrogen, the nitrogen-containing base. A is always going to bond to T in DNA, and C will always bond to G. And then next, you had to do 4.6. The main takeaway here are the differences between RNA and DNA. Chief among them, DNA has the sugar deoxyribose. RNA has the sugar ribose. DNA has two strands, and then RNA is just one strand of nucleotides. In DNA, you have the bases A, T, C, and G, okay, with A bonding to T and T bonding to A. But in RNA, the T gets replaced with U, which stands for uracil, and A bonds to U and U bonds to A. All right, mutations. Now, mutations are important because the state standards for 8th grade science have us learn about diseases. And some diseases are genetic, and so they come about partly because of mutations. They could be an error in the replication, just a mistake when they're copying the cells are copying the DNA. They could also happen spontaneously, caused by mutation or mutagen in the environment. No, you're not going to become a Ninja Turtle. You might get cancer or you might just die. Okay, but you're not becoming a Ninja Turtle. Don't try it. Okay. Examples include the carcinogens in tobacco smoke. They are a mutagen that cause lung cancer. They damage your DNA, and then your cell doesn't know when to stop replicating. And it just keeps replicating over and over again, and it forms a mass or a tumor. UV radiation can do the same thing with your skin cells. damage the DNA to the point they don't know when to stop reproducing, and they can lead to a skin cancer tumor. All right, so I'm going to move on from there. And 4.8 is the molecular genetics overview. You definitely need to know that the word allele means an alternative form of a gene. For example, there's the dominant gene for Huntington's disease that causes Huntington's disease, and there's the recessive gene that means you don't have Huntington's disease. In order to have a recessive trait, you have to be homozygous for it. So as far as I know, I don't have Huntington's disease, so I'm homozygous recessive for that trait. Yay, that's good. Dominant doesn't mean good and recessive doesn't mean bad. It depends on the trait you're talking about. So you should know the difference between homozygous and heterozygous, and we'll talk about that in the notes in a minute. You should know that if you're homozygous dominant for a dominant trait, then you have that trait. If you're heterozygous, you have the dominant trait. And if you're homozygous recessive, you have the recessive trait. Now, if you're heterozygous for a recessive genetic disorder, then you can be a carrier, which means you don't show the disease. The disease is not part of your phenotype, but it is part of your genotype, which means you can pass it on to your offspring. You cannot be a carrier of a dominant disorder because if you have the dominant trait, you have the disorder. All right. Let's see. And the only way to have a recessive trait, whether it's a disease or not, the only way to have a recessive trait is you get one copy from each parent. All right. Chromosomal disorders, these happen when something happens to the chromosomes. Usually this occurs during meiosis, which is the process that makes the sperm and the eggs. All right. So it could be a different number of chromosomes, or maybe a chromosome gets damaged. Let's see here. Down syndrome occurs when the zygote has an extra copy of chromosome 21. All right. Let's see here. And most embryos with extra chromosomes or too few chromosomes usually do not survive. All right. Lesson 412 is about the human genome. Another state standard is about biotechnology, and the Human Genome and the Human Genome Project and biotechnology in agriculture. These are all parts of that biotechnology standard. So a person's genome is all of his or her genetic information. You could also talk about the genome of a species, which is what the Human Genome Project did. And this species was obviously the humans. The Human Genome Project was an international effort to sequence all 3 billion bases that make up human DNA and to figure out the code for 20,000 human genes. And so, yeah, that was a lot. But they did figure out the sequence. I don't think they've translated all of the genes yet and figured out what all the genes do, but they have figured out the sequence of human DNA. So it's kind of like you know what all the words are in an alien language, but you don't know what all of the words mean yet. Gene therapy is where you take the gene that a person, maybe they're missing it, or maybe it doesn't work quite right. Like maybe someone who's born without the ability to make insulin. So then what you do is you insert that gene into their cells so that their cells can make insulin properly. And the way they usually do this is they take a virus, and all viruses do is inject their DNA, and then living cells are just like computers. They take that foreign DNA and they treat it like a computer program and they run it. And that computer program tells the cell to make new viruses. So they make more and more viruses until they rupture and release those viruses. All right. So, but you take the virus, you take out the bad parts, right? Throw those away. And then you insert the gene for, say, insulin. And then when you inject that virus into the people, it inserts the DNA for making insulin. And then all the cells that are get that treatment, they start making insulin. And then in agriculture, you need to know that a transgenic organism has DNA from some other organism. Example, there's something called BT corn. Most of the corn you buy in the grocery store is probably BT corn. And it had the gene from a bacteria inserted. And what that gene does is it makes a pesticide that kills the insects that try to eat it. Now, as far as I know, it only does this in the leaves. It doesn't do it in the corn itself. So it should be safe, right? I eat corn. It's one of the few vegetables I like, and I'm still here. All right. Ways that biotechnology can help agriculture. It helps improve crop yields so that there's more food produced. It helps make the crops resistant to things like drought. It makes them have more nutrients in some cases, improves the taste or texture. It reduces the amount of fertilizer or insecticides that farmers have to use. So those are all important aspects of using biotechnology. All right, so that's the end of the lesson, chapter review, lesson reviews, and I'm going to do the notes for today in a separate video. Stay curious.